We're going to start the final chapter today. So I know we have some things to go back to in vision, but we're going to talk about the chemical senses, starting with the taste system. The thing about the chemical senses is that they really are these gatekeepers. So they tell us where things are, whether it's food or mates, as we were getting into olfaction. Taste is particularly, so lets us know what's safe, what's dangerous, what gives us energy, what gives us salt, all of these things that we need. And so they're known as gatekeepers. With taste, we are bringing things into our bodies. And so taste, we have certain tastes that correspond to certain effects or outcomes. So sweet compounds, we tend to have an automatic acceptance response. Our gastrointestinal system prepares for getting something that is going to give us energy, and so it starts to get ready to digest. Bitter compounds, we have an automatic rejection response. And this is because many of the poisons that are out there are bitter, and they give the examples of strychnine, arsenic, and cyanide. Salty compounds tend to tell us about the presence of sodium, where if we have just sweated out a lot of sodium, or we need sodium, we've just peed out a lot of sodium, we might need sodium in our bodies, and so salty compounds can tell us that we are getting that. And of course, this connection is not 100% perfect, right? So aspartame, which is the artificial sweetener that we have in a lot of our diet drinks and things, it can suggest to the body that it's going to get the gastrointestinal system prepared for digesting and getting this energy. But it's a fooler, and it can actually, so it fools our system into thinking we should be getting energy when we're not going to. And there are other possible sweet compounds like that. Bitter compounds, as most of us drink coffee, that is something. Do we all have to do this kind of classical conditioning with coffee? Probably. It's pretty bitter, but it isn't any kind of poison. And there are lots of actually some of those vegetables that I tell you very early on, hey, these are really good for your eyes. Those green leafy vegetables like kale and broccoli and collards, those are bitter. And again, that's a huge fooler, right? Because they're really good for us. And yet a lot of people really don't like them because they have this bitter taste. And so we have a bit of a, ooh, yuck, bitter automatic rejection response. Currently, the 5 basic taste qualities that are accepted as basic taste qualities, which means we have receptors that respond to these taste qualities, are salty, sour, sweet, bitter, and umami. Traditionally, most of the research has been on salty, sour, sweet, and bitter. And I have in parentheses there that your authors have given us like a really good example. Sodium chloride is a really good example of something that is salty and just salty. Hydrochloric acid, something that is sour. Sucrose, something that is sweet. And quinine is bitter, where if we do these kinds of magnitude estimations like you see over on the right, we're just saying quinine, that's just bitter. And MSG, which they used to talk about as kind of salty, they now talk about that as umami, where umami was discovered and discussed in the 1990s, that we found that we have receptors for this kind of savory or meaty or brothy. It depends on who you ask exactly, but those are all good descriptions of umami. But so if we look at magnitude estimations for other things, if we look at, let's say, potassium chloride at the top, you can see that oftentimes when we're doing a magnitude estimation, people are saying it's very salty and it has some bitter components, but not so much sour and sweet. And if we look at sodium nitrate, it has some salty, some sour, and some bitter, and they're almost kind of equal. It has a bit of all of those, and they're not really very sweet. It's not very sweet. And so a lot of times we could have these basic taste qualities, we can do these magnitude estimations, and we can see how much do different compounds give us each of those tastes. And I have up there fats or fatty acids. This is actually a teeny bit controversial, as are heat and cool, which they suggest we're actually feeling those with thermoreceptors, but that maybe we do have taste receptors. There are a number of ways to discover what kinds of taste receptors we have, and so there's lots of research looking at these things. Fats or fatty acids, there are suggestions that it is something of the texture on the tongue that is telling us about that, and so it's more about touch, the touch of the tongue rather than taste. But again, it is a little bit controversial, and people have done research looking into maybe the idea that maybe fats or fatty acids are might be discovered one day as another basic taste quality. On the tongue, we have all of these different papillae, which have, most of them have taste buds. Filiform papillae do not have taste buds, so we're not tasting with those papillae. They are shaped like cones, and they're all over the tongue. And so we actually have this big blind spot, so to speak, in the middle of our tongue. And if you want to do this, one of my bio textbooks, I thought this was a fun thing to do. Dip like a cotton ball or a Q-tip or something into a salty or a bitter solution. Put it on the center of your tongue. Be very careful that it's right in the center. And notice that you don't taste anything when you move it over a little bit so it's touching the sides and it's touching those fungiform papillae. You start to get this sense of taste and potentially flavor, depending on what your solution is. And because if it's still touching at all, or if you move it so it's a big cotton ball and you're touching the center of your tongue still, it'll taste like it's coming from the center of the tongue. We have this capture as that touch system is pulling, so it feels like we're tasting things from the center of our tongues, but we're not. So filiform papillae, no taste buds. The rest of these papillae do have taste buds. So fungiform papillae, shaped like mushrooms, found at the tip and the sides of the tongue. We will talk mostly about fungiform papillae. I'm pretty sure that's when they're doing these counts of papillae or talking about different taste receptors, they tend to look at those fungiform papillae. But we also have a series of folds at the back of the tongue called foliate papillae, and then the circumvallate papillae that are shaped like flat mounds and surrounded by this kind of trench at the very back of the tongue. And so I'll go to our next picture to kind of show these. I will also say here that if we're looking at the tongue, or if we're thinking about where we're tasting from, all of the research I've ever seen is coming from the tongue, but we do have taste cells, I want to say on the soft palate, in our cheeks, and a little bit down the throat, but still, okay. So I have my information here of all the papillae except for filiform papillae contain taste buds, and the whole tongue contains about 10,000 taste buds. We don't have taste buds in the center of the tongue. Each taste bud contains 50 to 100 taste cells, which have tips that protrude up into the taste pore. And so they give us these magnifications. If we're looking at the front of those fungiform papillae and we magnify one of those papillae or papilla, we see the taste buds on there. And then we can magnify that and look at the taste bud, and you can kind of see it. To me, it looks like a little bit like cloves of garlic, except we have those somas in there. And then if we go and look at just one of those, you can see at the very top, there are there's a taste cell and its little receptors coming up. And what this is showing, if we look at the very top, that it looks like we have receptors for sour and salt. So sodium and hydrogen can just pass through ion channels, but that bitter and sweet are more complex. They are metabotropic. You don't need to know that, but they are a little bit more complex. And then umami is probably the same as bitter and sweet, but they haven't done as much research to figure that out. You might notice, so I'm going to say that there are about 50,000 taste cells. That is usually the estimate for humans. That does not multiply out correctly in what we just looked at. It should be 500,000 to a million. I don't know why those. And every book that I see does this same thing. If we have 50 to 100 taste cells per taste bud, and we have 10,000 taste buds, some people suggest maybe about 5,000, but it never really multiplies out. Sorry about that. I don't know why. That's That's what they say. But I'll do this little bit of comparison, and I will do so my word art is up there to remind me to say transduction. So this magical thing happens that the taste cell then can communicate to those cranial nerves, with different ones going to different cranial nerves. So good fun interspecies comparison, since I like to give those if I can. We have about 50,000 taste cells. They say chickens have about 24, so far fewer, which is why you can feed a chicken pretty much anything and they're going to eat it. And they taste things and they're like, okay, whatever. And catfish have about 175,000, and it's there on the, some of them are on the outside of the body. If you think about it, this makes sense. They are bottom feeders, and they're just picking up and tasting everything and spitting it back out, basically, to see what's safe. So they're a gatekeeper system. Okay, and transduction occurs when these chemicals contact receptor sites at the very tips of those taste cells, as we're seeing these cilia protrude into the taste pore. Here I have put this on a slide just because it makes sense to say it with wordy words and language. So from what we think of how this works, sodium and hydrogen pass through ion channels, which is creating that experience of salty and sour. Whereas receptors for sweet, bitter, and umami are these protein strings that cross the taste receptor membrane 7 times. On the previous slide, I mentioned metabotropic responses and said you don't really need to know that. You don't need to know this either, but it looks like, and it's kind of neat if you think about it, because that's what we saw with our rods and cones and those opsin strands, right, that they're crossing through that membrane 7 times. We see that here. We're going to see actually that kind of setup or mechanism in our olfactory system as well, in our smell system. So there you go. Transduction. If we look at the electrical signals that are transmitted from the tongue, we have a number of cranial nerves taking that information to the brain. And I'm going to start with this top one, the chorda tympani, and I'm going to basically say this is all you need to know. This comes from the taste cells on the front and the sides of the tongue, so from those fungiform papillae, and all of the research that we're going to look at that has to do anything with cranial nerves looks at the chorda tympani. Interestingly enough, I don't know if those other ones, I have no idea. I don't know anything about those, except that here they are. But they're in the book, and this is what the book says about them. But we'll stick with the chorda tympani. So the other ones, the glossopharyngeal from the back of the tongue, the vagus nerve from the mouth and throat, and the superficial petrosal from the soft palate and the top of the mouth. As we get to the main pathway for taste, those different cranial nerves are going to synapse in a place called the nucleus of the solitary tract, which is in the medulla or in the brainstem. So the medulla, which is in the brainstem, which we haven't really talked about very much, but some very basic survival mechanisms are in the medulla. And then we're going to go to our thalamic nucleus, which they have as the ventral posterior. Don't know if it's that simple. I'm pretty sure I've seen other things for taste. And then the primary receiving areas for taste are the insula and the frontal operculum cortex, or cortices, because the insular cortex is usually called a cortex. But so if we look at where those are in the brain, so we've gone through the brainstem up to the thalamus, up to our thalamic nucleus, we've done our sorting and filtering, and then it goes to this area. You can see that it is, it says 2 areas of the frontal lobe partially hidden by the temporal lobe. Honestly, I have seen these as being put in the anterior parietal lobes, the superior temporal lobes, the inferior frontal lobes. It looks like it's kind of, if you can kind of tell what's happening there. It's where these lobes come together. And so different books say different things, and I'm not going to ask. But there are two. And you can see why there are two, that they're kind of wrapped around this sulcus, this big fissure. So the insula and the frontal operculum.